therapeutic material. Yeah. As Sherajit et al., I'm at the Leibniz Institute of New Materials in Saarbrücken, and an institute for hybrid and biohybrid materials. So let's start by challenging you a little bit, or by exposing you the challenge that we all go through when we use therapeutics. And a very illustrative example is our eye drops. So whenever you take eye drops, which are very useful, and we are all very happy to use them whenever we need them. You have realized, I'm sure, that most of the fluid you put in actually gets lost, and actually it's not the fluid only that gets lost, it's also the drug that gets lost. So 95% of what you put in your eye is actually lost as soon as you put it, is wasted, and there is not much you can do about it. It's just we have an inefficient delivery mechanism. It doesn't have anything to do with the inefficient drug. The drugs are efficient, but we just cannot deliver them properly, and we waste a lot. And this poses, of course, it has an ecological concern, but it's also a concern for new drugs, in particular biopharmaceutics, when you think that they are more complex, also more expensive. So a drug that you put in your eye and you lose 95% definitely has to be cheap. So access of new drugs to ocular therapeutics becomes more difficult, at least via eye drops. Now, eye drops makes 80% of the delivery in ocular therapeutics. So we are talking about quite a large impact, and a problem for new drugs to come into to be used for ocular therapeutics. One possible solution, there are certainly more than one, but one possible solution it comes from the world of living therapeutics, and this world this community says, well, I mean, most of the drugs, or many drugs, we produce many biotherapeutics, and we actually produce them with organisms which are not that far away from the organisms we have in our ocular microbiome. And also in other parts of the body. So why not, instead of producing the drug outside of the body and then bringing it, packaging it and put it back into the body, why not just using the organism themselves and put the organisms in the body, where we know in many parts of the body we would have enough nutrients for them, and allow them to produce the drugs on site. Now, for that is in principle a very nice idea, or a appealing idea. Of course, we were talking about genetically modified organisms, not organisms that you have inside, organisms that, as in any cell-based therapy, would like to be defeated by your immune system. So it's a problem to put the organisms inside if it's not your own body cells, and it might also be difficult to deliver them. The advantage of using programmed organisms is that with all tools of synthetic biology, you can imagine that you can not only plan or program the delivery of a drug, you can also program the delivery of the drug in response to something. So the pharmacokinetics is no longer that needs to be done by the material as we are used to do with polymers. The kinetics would be done by the cells themselves. As I say, this is a nice approach, but again, we are putting up A, genetically modified organisms, B, into a body which is actually prepared for fighting against them. So it's difficult in some cases. The idea we're using, that we and others came in, and which is also now in the community, is well, if why not put in the organism in an encapsulated form? So why not encapsulate the drug, but organism, or encapsulating the biofactory of the drug? This is what we understand behind with the concept of a living therapeutic material. Is a hydrogel material, typically a hydrogel, so an hydrated material, a porous material, and for the whole part of my talk, it will be a hydrogel that can allow diffusion of molecules, nutrients, oxygen inside. So it can keep the cells alive, and it also allows the diffusion of whatever cells produce outside. In this way, we have the cells which we deliver to the body in a protected form, so they do not make contact with the immune system. This reduces the immune response, ideally. But the material allows the exchange of secretomes between the in our cases, in most of the cases, bacteria inside or drug-producing biofactories and the host. And now, of course, the matrix is an important part because it has to support, it has to control, but it also has to confine the organisms long-term if we want this as a long-term implant to treat, for example, chronic diseases. This is where such an approach would be most attractive. So let's go back to the case or to I introduced it before. Let's go back to the eye, and what would this mean for the eye? For the eye would mean, for example, that we could imagine an ocular device, for example, a contact lens, in which we could put bacteria inside or biofactories inside, and those would actually deliver the drug which otherwise we put on with the drops. And this is a beautiful material problem because on the one hand, you need to take care of all the specifications of the material requirements of a contact lens, which is actually quite high and engineered material to for you to be able to see and to wear it on the surface of the eye. And on top of that, we need to eat the same material or at least a material which fulfills all these conditions, also has to fulfill the specifications for sustained drug release. Now, those are different than the physical properties or optical properties that we need for vision correction. Those have to do with the organism. So the material has to be diffusive to nutrients, metabolic products of different sizes to be useful for drug delivery. It has to support the growth of the embodied biofactories. It had to retain the biofactories long-term, also in terms of biocontainments. And of course, it has to be processable at biofactory compatible conditions, which are not necessarily the ones that we use nowadays for producing contact lenses with the optical properties we need. So that's the challenge. That's quite a challenge. This is a multi-objective challenge, which, occupies us or keeps us busy, has kept us busy in the last years. Now, if you think about material selection for such an application, let's focus on the requirements that we need for contact lenses, for a living contact lens, for embedding a material. What we need is, ideally a material which is already approved for contact lenses. So we started with is polyvinyl alcohol. There are commercial available contact lenses based on cross-linkable polyvinyl alcohol modified with acrylates, acrylate groups. Ideally, if you want to make it compatible with the cells, you do not start from toxic precursors. That's for PBA is good because to make cross-linkable PBA, we typically start with long chains, which are also typically less toxic than short chains for mixing with the cells. It forms PBA. We can cross-link at low, or we can form stable hydrogels, at least as stable as a contact lens needs to be with a low polymer contact. This is also favorable for keeping the organisms alive. And it's also photocross-linkable, and it's photocross-linkable also at doses which are compatible with the living system. So it's an approved polymer for contact lens, which has also good properties in order to combine it with a living organism. And for the next slides, what we will see, we will be looking at PBA or mixes of PBA with with vinyl sulfone PBA, so that we can cross-link it also with photocross-link. This is the material base we use. Now we started preparing different hydrogels from PBA, and we came up with these compositions, just for you to have an idea of what kind of polymer we're dealing with. We're dealing with mix of PBA vinyl sulfone with concentration between 5 and 10%. Those concentrations give physical properties of relevance for a contact lens, which are close to the properties of the PBA, commercially available PBA-based contact lens in terms of water contact, in terms of oxygen permeability, water contact angle, transparency, refractive index, and also storage modulus. So this is good because, we are not far away from what it is already applied, so we can fulfill the material properties, but still we know that also under those conditions or with these compositions, these are also compatible with our cells. Which properties, additional properties, we need to check beyond the optical properties for our purposes? One is the permeability to large molecules, or to molecules in any way, but in particular to large molecules. You will see later on why. And what we see is that the compositions that we are dealing with, they are permeable to long molecules. So here we are looking at the release of polyvinyl alcohol of 130 kilodaltons, and you see that it takes some time for the polyvinyl alcohol to diffuse through a hydrogel. In this case, this hydrogel is around 1 millimeter thick, but it does diffuse out, and it comes out from the other side. So we can actually release large molecules with these hydrogels, and small ones, of course, as well. And yeah, it takes time. It takes days, which is good, because if we want to release something from a contact lens, we do not want to release it in seconds. So it's actually a good starting point. Also, proteins diffuse through proteins from 12 to 160 kilodalton. They diffuse, and they diffuse faster the smaller they are. So yeah, that's also what we would expect. Now, how do we design this contact lens? This is important. So we design this contact lens actually as a contact lens with two compositions of PVA. We have and we do this to decouple to some extent what the cells need and what the optical property correction needs. So our lenses are, we produce or we fabricate lenses which have, in which we embed a bio-ring. This is what you see here. This is this is the bio-ring, okay? And if you would see inside, you would see this is the ring, the blue part, and in the ring we have the biofactories contained. The material in the bio-ring and the material in the rest of the contact lens are slightly different. To be more precise, the material where the PVA composition, where the biofactories grow, has a little less solid content. So here we use about 5% PVA and vinyl sulfone, and outside we use 8%. We fabricate these contact lenses or prototypes in a two-step process. So first, we fabricate the bio-ring, which contains the bacteria, and then we mold, as contact lenses are produced also in the industry. We mold the rest of the lens so that the ring is embedded. The lens is transparent, you see here, and it's also flexible, has properties, mechanical properties, very close to your contact lenses. To test whether this idea could work, we decided to start with biofactories of hyaluronic acid. Hyaluronic acid is what you have in most of the drops you put in your eyes if you have dry eyes or dry eye symptoms, okay? You have a diluted solution of hyaluronic acid with different molecular weights. This is what you typically use. The more expensive the drops are, the higher the molecular weight of the hyaluronic acid is that you have inside. Yeah, for our first studies, we got actually a biofactory, which is Corynebacterium glutamicum. That biofactory was engineered to produce hyaluronic acid. Corynebacterium glutamicum is a soil bacterium, so it's around us. It's also a grass bacterium, so generally regarded as safe, and it's also a working horse of the biotechnology industry. That is included in the ring, and then we have the lens outside. So this is a lens which contains the biofactories. It looks like a normal lens. Outside in the ring, you would have the biofactories, and you zoom in. What you see is that the bacteria are contained, so the biofactories are contained in the ring. And they can actually not grow outside. And I will show you later on. And here you see the composition. So it's Corynebacterium glutamicum engineered, and PVA 5 and 8%. 5 is in the bio-ring and 8% is the lens. Now, yeah, does this lens really release hyaluronic acid? This is our target. And yeah, you see here that it does work. So this is a contact lens, as I showed you before, a hydrogel containing a bacterial hydrogel. in which we have incubated in medium over several days, actually 2 weeks, and you see, and we have measured on the supernatant if hyaluronic acid is produced, and you see that hyaluronic is produced continually up to at least 21 days. And within this material, with this composition, we see no release of bacteria. So it's also the matrix is also providing the biocontainment we need. We can also see that we don't know actually if, and we don't know yet, these are experiments which we are running now with natural tear fluid, but we actually do not know if the glucose concentration in the tear fluid is enough to keep bacteria active or biofactors active for a long time. So we could imagine also that in an application, from an application perspective, one could also feed the bacteria overnight in the care solution and then have them released during the day. And so we have checked this experiment. We have simulated this experiment in vitro. What we have here are two different type of experiments. In one experiment, we have kept the lens for during night in simulated tear fluids without nutrients. Okay, so it's only buffer. And in the other case, we have incubated the lens during night in simulated tear fluids and with medium, okay, which is more closer to what we really have in the tear fluid. And then we have done for several days. So we have incubated during night and measured or detected hyaluronic acid release during day. And it's very nice to see that if you do not feed the bacteria inside, then over consecutive days, the release of hyaluronic acid is dropping. Whereas if you feed the bacteria during night in your simulated care solution and you measure release during day, you can actually keep release also more or less contact over several days. So this is encouraging because in the case that we would not be able to, in case that the feeding conditions or nutrient conditions in the eye would not be enough, one could even do it over day and have a reusable lens which you can self-replenish in terms of drug content overnight. Now, of course, the question is, does it really work? So if we deliver hyaluronic acid, does the hyaluronic acid which is delivered really contribute to friction, to improve or reduce friction properties and improve lubrication of the lens? This is what we show in this experiment. So here we have a hydrogels, as we saw before, which we have filled with different things, either with nothing, okay, so the empty ring, or with a ring which is containing, or with or we are testing it with the liquid or the release hyaluronic acid, which comes from wild type bacteria. So these bacteria have not been engineered to produce efficiently hyaluronic acid, and ideally nothing or not much of the rest. These are the wild type, and also with our engineered bacteria. And what we look at here is the coefficient of friction of that lens in the supernatant. Now, when we have the hydrogel, we see no difference over time because we have no factories producing anything. The gel is also stable, so coefficient of friction remains over time. If this is incubated with this is measured with the supernatant from hydrogels that have been contain bacteria, then you see that the friction goes up, and this has to do with all the other products which wild type bacteria would secrete. So they deposit on the contact lens. This is also why we have to clean the contact lenses, and we actually see that the friction goes up as the deposition or as the concentration of the secretome of the bacteria go up, also the friction goes up. And in the case of the lens, where we secrete that contain the bacteria that secrete the hyaluronic acid, the friction goes down, which is what we were, of course, looking for. So these are very promising examples. These are very promising study to show that, whatever is secreted from the lens can contribute to reduce the friction of the surface of the hydrogel. Yeah, when we reported this, we got a lot of media coverage on ophthalmology journals, thinking that this or stating that this eye would be the solution to dry eye problems. We are definitely not that far. We would like to be that far, but we're not that far. But we're indeed following up on this idea and trying to push forward the technology to make self-lubricating contact lenses. So we are in the working on the technology transfer step. Not only for self-lubricating contact lenses, but actually this is more or less this is quite a general project concept, and you could imagine also delivering many other type of things, not only hyaluronic acid. Here you have an example. These are similar gels, but actually, delivering containing a biofactory of a natural anti-infective, which is also delivered. This is why they are colored. So you could imagine this approach to work also with other biotherapeutic molecules, not only with hyaluronic acid, and this is what we are currently working on. You can also imagine this approach working for many other type of drug delivery devices, not only for the eye. And we also work on printing technologies, on microencapsulation technologies, also on electrospinning. So and this would end up having fibers or capsules or meshes, which you could also implant, and in which you can use to generate environments for the bacteria in which they can produce the drug, and also they can be implanted. You'll see some of them later on in the talk. Okay, so if we go back to the concept of the living therapeutic material, I hope with this example first, I could show you that it seems possible. Okay. But of course, the question of a release, a programmable release, is important. In the case I've showed you before, the release occurs passively. And I show you how it occurs passively. But this is the first approach, okay, to programmable pharmacokinetics, and the way and why it occurs passively, or what is the passive regulation, is actually the mechanical properties of the hydrogel. So those are tuned to actually control growth, and this is known from biophysics. So when you think that when you prepare the ring in case of the contact lens, or any encapsulated organism or bacteria, if you put them in nutrient and the matrix is diffusive, would try to grow, okay, would try to multiply. And to grow, they have to, they apply the tubal pressure, they have to divide, they have to become two out of one. And for that, of course, as they grow, they need more space, and they need to deform the material. Now, if you think that you are having a cross-link, chemically cross-linked PVA, then of course the material will allow this to be deformed. But as it is deformed, it's applying a compressive force on the colony, which is growing. It is this compression force actually which slow down and restrict growth. Now, this is a biophysical principle which is known and which has been also tested, not in hydrogels, but in cavities and so on. So this is also the one that we have been used for in the lens. But let me show you how it is in a different material, which I will use for the next case I'll present you. So we wanted to test whether this is really the case and how far we can program passively the growth of bacteria and the retention of bacteria also inside a hydrogel with a pluronic hydrogels. Now, pluronic, this is a different system. Pluronic is forms, as is the triple copolymer, forms micellar hydrogels, form physical hydrogels at certain concentration, actually quite low, and these are thermoresponsive. So you can actually, they are the temperature, the transition temperature at the concentrations we work around 23% is around 15 degrees, and or 15 to 20, no, here is 22, but this depends also on the molecular weights, but it's below room temperature and below also body temperature. So you can process them actually below that temperature as a fluid. You can mix them with the bacteria, and then you can let them solidify and form a physical hydrogel at body temperature, or afterwards at room temperature as well. You can also mix pluronic with pluronic diacrylate and then form a system, form a physical hydrogel in which you can impose a chemical cross-linking as well without disturbing the physical order. By that allows you to make hydrogels which have in which you can control the physical properties. You don't change the solid content, you only change the amount of chemical cross-linked on top of the physical cross-link. And you can generate gels, which you can also mix with bacteria, you will see in a second, with polymer, with shear modulus between 10 and 50 kilopascals, and also polymers which have a critical yield stress between very low one and 10 kilopascals. So this is important also in terms of deformation, or the possibility of the biofactories to deform them. Now with these hydrogels, if you put bacteria in these hydrogels, you see the following. So if you put them in the physical hydrogel, the one with the lower shear modulus, you would see that bacteria actually grow very fast and fill the whole hydrogel. They actually outgrow the hydrogel as well, sorry. Whereas if you do this with the hydrogel, which is heavily chemically cross-linked, bacteria also grow, but also they grow much less. You see here they form actually like small balls, small colonies, and they stop growing. They would never outgrow the hydrogel. I run the videos again so that you see the difference. Here they grow out, here they stay in. If we quantify this, if we do this in thin films and we quantify growth, this is what we observe. We observe the highest colony growth in the hydrogels with the lowest covalent cross-linking, and we have the lowest. We quantify the lowest growth in the hydrogels with the higher cross-link, and we see no outgrowth, and this is important. That means we can generate, similar to as I told you in our gels, in our contact lenses, we can generate core shell composites or core shell materials in which we have we can generate, so we can leave the cells inside in one type of material and have the other type of material outside which contains the growth. If we quantify growth inside, then we see that also we can, and this keeps viable and also no leaking over months. We can then go to check how growth runs, and we see that growth goes actually happens in the bacteria with intermediate concentrations and also protein production. So we can by that generate systems in which we can maximize production at the certain polymer conditions and minimize growth or maximize growth depending on which composition we use at others. So all these parameters we can control by that. We can control growth, we can control production, we can also control outgrowth by changing the compositions. One question, Sergey. I assume that I had 40 minutes for the talk. Am I right?
Well, you have 1 hour, but it's up to you how long you are free
then
to
we are
to
fine.
go.
I was just making sure that that was the right time. Okay, then I'll continue for the next 15 minutes. That is the passive, how we can passively control growth with mechanical properties of the polymer. We can control the growth and we can control production, and this is what we use for our contact lenses. Now, can we control actively? So can we make a responsive system? And as I said at the beginning, we can do that if we use or if we program the organisms. We can, for example, program organisms to do synthesis or to synthesize certain drugs in response to light. This is what optogenetic people do. And here we have one example from our collaborators. We have a bacterium which is able to produce a drug. This is the oxybutyllacin. It's also a colored drug which we can see with our eyes when the culture is irradiated. Here you see how it looks like. So this is the hydrogel. This is the hydrogel with the bacteria, and this is the hydrogel with bacteria which has been exposed to light and becomes colored, which is a sign of the production of the oxybutyllacin. And of course, if you have light, you can then actively control drug production. You can do this on demand. You can also do it as a function of dose. You can keep this long term with these organisms. You see it here. It goes over weeks. And you can spatially define also where you produce the drug if you focus your light dose. So the properties you would expect from a light responsive system. Now the issue in that problem now is that if you want to do this inside the body, you of course it's not enough to have organisms that respond to light. You need also to try to bring the light inside the body, and so we have been working now on devices that would allow transport of light and also control of the organisms and encapsulation and control of the organisms in a single device. These are our bio-optical or our living fibers for photopharmacology. So you can imagine a fiber like that. This is actually a thread, a fiber with 3 layers. The layer inside is an optical fiber. It's a special optical fiber. It's not only an optical fiber. It's an optical fiber which also has segments that can outcouple light because they are in there. We have particles scatterers for outcoupling light, and also other type of particles I will show you afterwards with plasmonic effects. In the intermediate layers, we have the biofactories, which are sensitive to light. So we could, with this approach, we can, with these devices, we can bring light, we can outcouple light and excite locally also the factories in the intermediate layer. And we have an outer layer, which is the biocontainment layer to hinder the outgrowth of biofactories. Now, what's important here to note is that we are using actually almost the same material for all 3 layers, or the same ink. We are just adding scatterers or cells to make it, or eventually also changing the ratio between the pluronic and the pluronic diacrylate to optimize the properties for the function of the layer. We can, pluronic is a polymer that you can print or extrude quite good. It has shear thinning properties, is actually very handleable for printing. Eventually also many people are using it also or you are using it as well. This is, so we use coaxial extrusion printing to produce these fibers. Here you have an example of the simpler one. So you have, we have a core shell, a coaxial extrusion nozzle. We have a core shell structure where the bits inside, you see that we have also segmented them. I'll come to that in a second. The parts inside the core is containing the bacteria. Outside we have the biocontainment layer, and we are using the two different types of pluronic mixtures. Same solid content, just by changing the ratio of acrylated and not acrylated ones, we can change the properties. In this polymer, also bacteria grow inside, they grow for a long time. We can also, if we do the printing and the devices, if we print the device correctly, we can also keep bacteria growing inside for a long time without seeing any leakage. And the pluronic is not only a good material for printing, it's also optical. It's an hydrogel with acceptable goods or with good optical properties, at least for exposure inside the body. Is transparent or can be transparent depending on the formulation at visible wavelength, and you can tune the refractive index by the solid content of the polymer. So that allows you also to make core shell fibers in which you get the maximum total internal reflection. To make now, we can print pluronic, as I said. We can to make the side emitting fibers, as I told you at the design at the beginning. We need to use a multi-material printing approach in which we can combine, we can use, we alternate and inks. The ink that has wave-guiding properties, this would be the pluronic, and the ink that combines the pluronic plus scattering nanoparticles, it would be the scattering ink. And you see, this is an example. These are these 2 inks, yes, with color to see them, and you see that we can actually alternate them in depending on the switch, on the switching between these 2 pistons or these 2 containers. For the printing, we can switch them, and we can get continuous threads with alternating composition. This is the product out of that. So this is a waveguide. The parts which are not red are the waveguiding ones. The red parts contain scattering particles. This is why you visualize them. And these would be the outcoupling ones. And we can, as we saw before, we can change the period. We can change the length of the scattering elements depending on the switching time between the two inks. This is a real wavelength, which is also coupled to a laser. So here we are coupling the laser from this side. You see, this is the waveguided segments. These are the outcoupling segments, which we can place as we want. And here, of course, is the end of the lever. So the waveguides are physical objects. You can couple light. You can use them. The beauty of this fact that we can print segmented fibers or we have this multi-material printing process is that with that we can control the length and also the profile of the outcouple light. We want to outcouple where the bacteria are later on. And depending on the switching profile, we can either outcouple over a long segment or outcouple in short segments as we want. Here you see the outcoupling, the intensity of the outcouple light along the fiber. If we don't use scattering particles, but we use plasmonic particles, then we can do a similar. We can achieve a similar. We can use the same approach to achieve fiber with photothermal properties. Here you see this is a fiber which has segments of containing plasmonic particles, and they can be heat up with lights and also produce localized heating. As also you can actually also change the laser power and control how much you heat, and you can do that also in cycles and repeat it. So you can stop heating and activate heating again with lights. So where are we in the process? Now we have the waveguides, we have the outcoupling elements, we have the possibility to print also the bacteria and to keep them alive. And this, now we have to put them together. So the nozzle is now a bit more complicated. It has then 4 inlets. The ones in the middle are for the segmented waveguides with scattering or thermal or plasmonic elements, and with the waveguiding elements. Then we have the one on the right. This inlet is for the bacteria ink or for the bio ink, and this inlet here is for the protective layers. And we can prepare these layers with these waveguides with the 3 layers to make the system work. Here you can see the layer, the middle layer, the inner layer, sorry, with the side emission, the core layer, and here you see bacteria colonies outside which have been grown after activation. And if you would look inside the fiber, you would see this is the core layer. So this is the waveguide. This is the intermediate layer in which the bacteria have grown, and this is the biocontainment layer outside. This is how it looks like. And we are working now on the localized heating. We are not there yet to get it as localized as we would like it, but you see here that it works. So here you see a plasmonic element, for example, and here you see also that we were able to excite the bacteria that are able to produce a red fluorescence protein outside. We do not get the localization also because we do not have controlled temperature around, which is what we're working on. But the principle should work. So to summarize, this is this would have been the approach to get active response or active delivery or active control of the pharmacokinetics with light or with temperature using a living therapeutic material. The few last minutes I want to use to introduce to the difficulties of using such materials also for application, and the fact that if you have a living therapeutic material versus having a non-living therapeutic material, is that in a non-living therapeutic material, you can measure delivery, you can measure the delivery of the drug as it degrades or as it swells in different conditions. And you will expect that the material, if the conditions don't change, also the function you get doesn't change. And this is different in the living therapeutic materials because you have living organisms inside which can change depending on the conditions outside. So we need a huge amount of experiments to be able to understand, predict, or well, first to understand and quantify, and then eventually also predict at the end how do all these devices actually work. And we have design factors which affect them, but we also have internal factors, is the evolution that the bacteria could have, or of course also of the material in response to the evolution of the material, the bacteria, and then external factors which will change also the behavior of our biofactories. That means that we need to think a little bit better how to perform the experiments. We cannot perform the experiment with the biofactory alone. We have to perform the experiment with the living materials, which means that we have to get a type of array formats in which we can reproduce these living materials, and so that we can also go for high content screening or also high throughput screening. This is how we are doing it now. So we are encapsulating our colonies in between two layers, simulating what we have in a lens, simulating what we could also have in a fiber. We do this with slot die coating, so we produce tins of the polymers. We can also spot or even deposit the other material inside. We can die coat on top. We can generate this kind of substrates which have a well format, have a well format type, and then do high throughput or at least medium throughput screening, which of the different factories as in a function of different parameters. This is here you see such a microarray in which in the middle we have the colony. The colony is not close to the borders, it's not close to up or down layers. This is also important. We can do this also in transverse, so that we can also expose them to asymmetric conditions. This is also important. So this is something in which we are investing quite some time now to try to design formats for the analysis of living therapeutic materials that allows us getting more data. You can do microscopy on them, so you can see how you can quantify and with different methods, also with imaging, how colonies grow in time and as a function of nutrient conditions or other conditions. You can also analyze and visualize the individual colonies. And by that, you can follow, you can take also supernatant from different conditions. You can analyze also supernatant to see the behavior of the factories, how vital they are, what they produce, what is the nutrient condition status, pH status, and so on, and do that as function of many different parameters. Okay, so with that, I come to the end. This is a summary of what we have seen. I introduce you to the reason why thinking about new forms of delivery is important from a sustainability perspective, but also from a perspective of being able to use more advanced drugs, which typically are also more expensive because they are more complex. I presented you our case of a lubricant or a hyaluronic contact lens, which delivers hyaluronic acid as a lubricant, as an example of such a device. We then went through to thinking that delivery is not alone. We need control delivery, and that we can do passive. The control in this type of devices, we can do it passively by mechanobiology, so by adjusting the mechanical properties of the matrix that contains the drug biofactories, or we can do it also actively if we program the biofactories. In the future, we might also be able to program both material and biofactory, but we are not there yet. But at least with the light-sensitive biofactories or temperature-sensitive biofactories, if we combine them with a device that allows us delivering light inside the body, this is also possible. This is where I presented you the living optical fibers. And finally, I introduced you to the problem of challenge of developing such devices, in particular if we try to transfer them or translate them into clinical applications, because they need a very extensive preclinical study, which is difficult to perform if we are working with devices, and which means that we have to also, in a way, find ways to how throughputize the devices in mini devices so that we can test in many different conditions before to have a good amount of preclinical tests before we can go in vivo. Yeah, that would be all from my side. Just to inform you that this type of work is performed in collaboration also with other people at the Saarbrücken campus. So we together with the Saarland University, we have the Leibniz Science Campus Living Therapeutic Materials, where we work on these approaches, not only on the eye, also on many different organs. So if you are interested in that, you can also visit our websites. And with that, yeah, I thank my group, for all the work that has that contributed to, of course, to what I have presented to you. Our cooperation partners in Saarland, in Leibniz, and also outside, and you, of course, for your attention today. Thank you.